University, where he studied androgen receptors with Cindy Moranti. Afterwards, he did 2 short postdocs at Johns Hopkins, one in the Department of Urology with Kenneth Pienta, and the second at the Cancer Center with Drew Pardal, which is the same postdoc, same lab I did my postdoc in all those years ago, which makes us scientific siblings. Now, he's an assistant professor at Johns Hopkins in oncology in the prostate cancer program, where he has his own lab and studies therapeutic resistance, metastasis, and immune cells in the prostate cancer microenvironment. Dr. Zarif is a member of several programs at Hopkins and an adjunct professor at Morgan State University. He has many nice manuscripts, is well-funded, and has mentored many trainees. He has a number of honors, and I just chose a couple to tell you about that I thought were the most interesting. In 2015, he got the Martin Luther King Jr. Award for Community Service. This recognizes his outstanding commitment to volunteer service. In 2016, he was recognized with the Prostate Cancer Foundation Young Investigator Award. This award identifies a cohort of leaders who keep the field of prostate cancer research vibrant with new ideas. And in 2018, he got the Howard L. Cornish Humanitarian Award from the Morgan State Alumni Association. And this was for mentoring college-bound high school students and for his research. The title of Dr. Zarif's talk today is Targeting M2 Tumor-Associated Macrophages to Overcome Tumor Immunity in Metastatic Castration-Resistant Prostate Cancer. Dr. Zarif, thank you so much for sharing your research with us today.
Again, thank you for the introduction. I'm honored to be here virtually and to share our work.
Right, it's all you.
All right. Thank you for the kind introduction again. Let's see. Let me just enlarge this. Everyone can see my slides? Great. So once again, thank you so much, Dr. Slansky and the organizing committee for allowing me to talk about our work today. This work is what our lab is working on. I'll also talk to you a little bit about some of our clear cell renal cell carcinoma data that was just published July 31st. So for those who are not in the field, I just wanted to share a little bit of the statistics about prostate cancer. It is one of the most commonly diagnosed cancers among American men. Nearly 175,000 new cases each year. It's initially treatable, but when it recurs, it can recur due to a lot of different resistance mechanisms. Some of note involve the androgen receptor mutations to enzalutamide and to other therapies. And then cells can eventually lose the androgen receptor and become resistant. They don't have the androgen receptor, or they have a variant 7 of the androgen receptor that does not respond to AR antagonism, as well as the amplification of MYC and PIK3CA, et cetera. Close to 32,000 deaths each year in the United States. And since we're in the era of addressing social injustices in our country, I will say that Black men are disproportionately affected by this disease and also are not readily recruited to clinical trials for prostate cancer, although the evidence in genetics suggests that African-American men have genetic differences that allow their prostate cancers to present a little more aggressive than men of direct European descent. Overall, the tumor is traditionally considered immunologically cold because of the paucity of CD4 and CD8 lymphocytes in the tumor at time of biopsy or resection of the prostate. Prostate cancer's common designation, final destination, I should say, is the bone, where it forms bone on top of bone. So it has osteoblastic phenotype. So it has this osteomimicry when it goes to the bone. And this is what I'm showing you here is a technetium-99 bone scan of one of Kim Pienta's patients, where you can see bone metastasis to the rib, to the spine, to the patella, as well as the shoulders. And this is what kills men each year. It's not prostate cancer diagnosis, because you can remove the prostate and it may never come back, or you could be on antagonists for long periods of time until your PSA is lowered and it never bothers you again. But for men who present with metastatic symptoms at time of diagnosis, it tends to hover around 33,000 men per year. And it's, again, a huge medical burden in the United States. And just think about this. It's 30,000 men per year. COVID has trumped that number already, and it came around in February, right? So castrate-resistant prostate cancer is one of the cancers that's resistant to checkpoint blockade as a monotherapy. And so one of the first two papers actually to come out were by Suzanne Topalian here at Johns Hopkins School of Medicine, as well as Drew, Dr. Slansky, and I, former mentor, where in this New England Journal of Medicine paper, the Topalian group had 17 men who had castrate-resistant prostate cancer, and 0 responded or had any positive responses to anti-PD-1 as a monotherapy. And so the field is looking to try to combine checkpoint blockades with other antagonists. There's one paper from Pam Sharma's group that came out in Cell, I think last week, that had some, I think 25% of men responded to IPI and Nivo combination therapy. But that's still a minority of patients. So the field is starting to look outside of lymphocytes, which is kind of sacrilegious if you've trained in the Pardo lab, because the Pardo lab is a T-cell lab. But the cells that I've been interested in since graduate school were macrophages. And they are part of the innate immune system. They're not a part of the adaptive immune system. And unlike lymphocytes, these cells are resident in practically every organ in your body. And then you can have monocytes that go to that site and become macrophages. So it's a little different than the adaptive side of things. And so this is just a cartoon of looking at not all, but several organs of the body. So the microglia of the brain are the resident macrophages of the brain. Kupffer cells are the resident macrophages of the liver, and the like. Again, because lymphocytes or checkpoint blockade is at the forefront, and the minority of patients are responding to it as a monotherapy in most cancers, there's a growing role of appreciation of myeloid cells in tumors. And so this is another cartoon from 2 years ago, where they just analyzed the immune microenvironment of several tumor types. Prostate, unfortunately, is not here, but I'll get into that. But bladder, breast, bowel, stomach, and lung. And you can see that the largest amount of cells that are in some of these tumors are macrophages, and not gamma delta T cells or B cells. What do we know about macrophages? So they are a heterogeneous population. Again, they're a cornerstone of innate immunity. They can be either resident macrophage and then differentiate or polarize into classically activated or M1 macrophage, or alternatively activated M2 macrophage. They have opposing roles. Or the cell can arise from a monocyte that traffics to an organ, differentiates into a macrophage, and then polarizes. So there's two types of ways that you can get a macrophage into a tumor microenvironment. And the M1 macrophage is more pro-inflammatory. is known for tissue damage and killing pathogens, and the M2s are anti-inflammatory, have a Th2 response, and promote angiogenesis. And these cells are potentially interconvertible, and this is something that we're exploring. Data from the Miriam Murad lab a few years ago showed that there's even diversity among resident macrophages. So this is just a Tisney plot showing macrophages, and what they ended up finding was when they profiled them using flow sorting and Tisney, is that there are about 15 subsets of resident macrophages in the lung. So we're still, learning a lot about macrophages, even though they were discovered by Michnikov in like 1896, and he was one of the first people to win a Nobel Prize for this work. We're still learning a lot about macrophages. Here are some of their functions. Again, just a cartoon. So they have tumor promoting functions where they can make EGF. They can lead to tumor invasion by making matrix metalloproteinases and cathepsins, such as cathepsin L, which I'll talk a little bit about. They can lead to angiogenesis by making VEGF, which we've shown using multiplex cytokine analysis, and so have others, as well as trafficking with CXCL8 or IL8, which Chuck Drake studies. And immune suppression, so they know how to be kryptonite to lymphocytes. If lymphocytes are CD4s and CD8s are supermen, these types of secretions are kryptonite to those cells. They don't proliferate well in the presence of some of these molecules. And then finally, M1s can have more of an anti-tumor effect by upregulating or secreting NOS, as well as recruiting cells to tumor microenvironments by STING or IL-12, which lots of people have studied before. So this is my lab's ethos and hypothesis. Yes, this is posted in the lab because everybody in the lab studies some aspect of this cartoon right here. And that is myeloid cells and M2 TAMs are evil incarnate, and they promote therapeutic resistance in prostate cancer metastasis. And we have data substantiating a lot of the parts of the cartoon. So we've co-cultured prostate cancer cells, PC3 epis, with monocytes. These monocytes end up becoming macrophages and promoting and lead to these epi cells becoming EMT. When we put these cells into thymic nude mice, these cells are more aggressive and metastatic faster than the PC3 epis. We do get some bone lesions. Unfortunately, they're osteolytic, which a prostate cancer cell, PC3 cell is. But in patients and in the syngenetic model, I'll show you that we do have osteoblastic lesions. There's another quagmire, at least in the tumor biology of prostate cancer, where tumors can go to the bone and remain quiescent for a period of time. And so you can't even detect them by technetium-99. And there are some PET PSMA agents that are allowing us to see tumors that are not visible by technetium-99. In any case, these METs end up showing up years later. And the thought is, at least by Jeff Pollard and others, that these tumors are devoid of TAM infiltrate when they first metastasize and settle in the bone. And once they recruit more myeloid cells, either from the bone marrow or from the circulation, then they form micrometastases and then frank metastases. And this leads to bone growth on top of bone and nerve compression syndromes and pain and cachexia and subsequently the death of the patient. So to study macrophages, you need a good model. It's really to study any cell. And so when I first started out as a fellow, we were reading a lot of literature about how to differentiate M1 and M2 macrophages from primary cells. And every time we tried these different protocols, there was ambiguity in our results because we had dendritic cell contamination. And so we came up with a method to differentiate monocytes, which are CD14 positive, and get homogeneous populations of M1 and M2 in an effort to learn what ideal and novel surface markers may be expressed on these specific cell types so that we can target them in the tumor microenvironments, particularly prostate cancer and probably others. And so I have in these orange boxes here what we did. So we get leukopaks from Johns Hopkins Phoresis Center. We do a phycol spin, which is standard, do a bunch of washes, clean it up, red blood cell lysis, and then we use magnetic beads from Miltini to get the CD14 positive cells. Then we differentiate these monocytes into macrophages. So for M1s, we use GM-CSF for 5 days by itself in RPMI media. We replenish the media in the GM-CSF and add lipopolysaccharide, which is a toll-like agonist, interferon gamma, and IL-6 to stimulate STAT3 phosphorylation. We let that go for 4 additional days. So this is a 9-day cultivation method, which consists of a differentiation period and then a differentiation period and then a polarization period for 4 days. We do the same for M2s, where we use MCSF for 5 days, replenish that on the 5th day with new media. That gets rid of detached cells, which are mostly dendritic cells, because dendritic cells can be activated similarly. And then we add IL-4, IL-13, and IL-6 for 4 additional days. And what we found is that these cells had high levels of mannose uptake to M2s compared to any of these other methods. Mannose is on the M2s. It's the mannose receptor, specifically CD206. And then for the M1s, we looked at lots of cytokine secretion, multinucleated giant cell formation, and the like. So I won't go through that whole paper because that paper is long. So from there, we wanted to identify novel surface glycoproteins that were unique to each of the macrophage subsets. And so we worked with Wei Zhang and Wei-Ming Yang here at Hopkins, and they developed a method called solid phase extraction of end-linked glycopeptides, or SPEG. And so they ran this in their lab after we gave them lots of cells from healthy donors. Then the orbit-trap liquid chromatography tandem mass spec, followed by the proteome discovery software using Sieve. And we looked at differential expression of glycoproteins on the cell surface of these myeloid cell types. These include the monocytes. So these are all patient matched. So we had monocytes. We differentiated monocytes to M1s and M2s, but we had a control monocyte from that donor. And so we did 3 different donors 3 different times. And we came up with these data. And so we saw that there was a different glycoproteomic signature among the, when you compared the monocytes to M1s, and that those differences are shown here on these edges here, as well as the monocytes compared to M2s and M1s to M2s. So we knew that the cells had a fundamental difference on the cell surface. And then we wanted to look for some of the top hits of those differences. So which of these proteins at a 1% false discovery rate was highly glycosylated. Now, biomarkers are typically highly glycosylated at an asparagine residue, such as PSA, CA9, and the like. And so one of our top hits was the mannose receptor, CD206 is also what it's called. This is the mass to charge ratio, showing that the asparagine residue is in red. And then when we compared the PBMC M1 and M2, you can see a nice peak at the retention time of about 41.75 from the mass spectrometer. We also found other proteins that were highly enriched that we're currently investigating. We chased after the mannose receptor for reasons that I'll tell you on the next couple slides. But the other hits were integrin alpha-3 cathepsin L, which I've already said is important for the tumor biology or the tumor microenvironment and what the macrophages bring to the tumor microenvironment. Legumine, sodium hydrogen exchanger 7. And so since we had CD206 as one of our top hits, we worked up a antibody with the lab of Angelo DiMarzo here at Johns Hopkins School of Medicine. He is a world-class GU pathologist who studies MYC. And this took us a while, actually a couple years to do. And so what we did is we took about 200 TMAs for men who had radical prostatectomies between 2009 and 2013. And so we had those TMAs. We performed immunohistochemistry on those TMAs to look at different stages of prostate cancer and relative to normal prostatic tissue to determine whether 206 expression in these tumors increased as the diagnosis got worse. So is macrophage infiltration, M2 TAM infiltration in prostate tissues a prognostic marker? And so these are images from the paper. So this is normal prostatic tissue. This is a Gleason 3 + 4 = 7, where you can see nice CD206 stains, as well as you start to see the breakdown of luminal structures that you typically see. This is a lymph node MET, where you can see TAM infiltration here, and then this is a distant MET. And so with GU pathology fellows in the DiMarzo lab, we wanted to look the mass area ratio of the mannose receptor. The gene name is MRC1. And what we can see is that as the disease progresses, there's a crescendo in CD206 infiltration into the TMAs that we were able to score. We also looked for 206-68 stains. So CD68 is a pan marker for macrophages, so it stains both M1 and M2. We wanted to look for this by IF, and we were able to see, and this is just the merge, where we were able to see that there are TAMs that are M2 or CD206 positive in the tumor microenvironment, because we didn't do that. We didn't do that here. And we also have a rapid autopsy legacy program. And so we've done about 30 cases here. All of these cases are consented and coordinated by Dr. Jody Hooper and Dr. Angelo DiMarzo, who Dr. Jody Hooper runs the legacy rapid autopsy program. And when we performed, tissue dissociation, tumor dissociation using the octodissociator on freshly isolated tumors from rapid autopsy, and then perform a simple CD206-163 dual stain of the sample to look for TAM infiltration, we're always able to see that there's a high percent of dual positive CD206-163 stain. 163 is the scavenger receptor. It's also another canonical marker of M2 macrophages. When we look for M1 macrophages using co-stimulatory receptor CD86, as well as CD206, because some M1s can have very low levels of 206, we see a paucity of these cells in these rapid autopsy samples. And finally, when we look at the terminal site of prostate cancer, which is the bone, we're able to see CD206 stain in the bone. And that's shown here alongside the IgG control. And so we worked with a group, Riptide, as well as scientists from Tuskegee University, which is in Tuskegee, Alabama. And they, specifically Jesse James, who's at Tuskegee, developed this peptide called RP182 that binds to the mannose receptor in the fibronectin domain shown here. And this was published in February in Science Translational Medicine. And so we worked with this group to see if we could kill our M2 macrophages with this peptide. And what this peptide does is it binds to the fibronectin type 2 domain of CD206 and causes conformational change. This CD206 is then endocytosed, and for reasons we're still trying to understand, it leads to activations of caspases, specifically 7, 3, and 8, which are execution of caspases for macrophages. And so I won't go through the entire paper, but what I will show is that this peptide decreased the viability of human M2 macrophages compared to M1 shown here, as well as the murine macrophages that we were able to isolate from bone marrow and differentiate. And then we looked at a fluorescence of caspase 3 and 8, and we're able to show that this does activate those caspases, and they're statistically significant. And then we looked at the localization of RP182 after 2 hours. So we see that it does get inside the cell. And this was in collaboration with Udo Rudloff at the NCI, as well as, again, groups at Tuskegee University. We also studied clinically localized treatment-naive clear cell renal cell carcinoma. I know this is a prostate cancer talk, but we have also begun analyzing some of these samples that are treatment-naive. And what we are also seeing is that there's a high level of myeloid-infiltrating cells into these tumors compared to the normal adjacent tissue, which we get a lot more because the kidney is typically a bigger organ than the prostate. Our prostate is about the size of a walnut. And this was led by Debebe Tedros, who's an MD PhD candidate in my lab. So 2 other ways that we are planning to target M2 TAMs is one, glutamine-dependent signaling and targeting, and this is work that is ongoing in collaboration with Jonathan Powell and Barbara Slusher, and then finally PI3 kinase signaling. So targeting metabolism. So in the first paper I showed where we figured out how to differentiate CD14 positive monocytes into M1 and M2 macrophages, there was another paper that came out around that time in immunity from Agios Biosciences. And what they did is they took bone marrow derived mouse monocytes and differentiated them into M1 and M2 macrophages in the absence of glutamine. And they saw that the cells that they tried to differentiate into M2 macrophages actually became more M1-like in the absence of glutamine. So we saw that paper, and I said we should repeat that with the human samples. And that's exactly what we did, and we saw similar findings. And so shown here in orange is CD86. It's typically ascribed to being a M1 marker. CD206 in orange, if you see that back shift, again, this is on a log scale, so there's a decrease compared to blue. There's a decrease in CD206 stain in the absence of glutamine, shown here in orange. E-cadherin, yes, macrophages have epithelial cadherin, even though they're not epithelial cells. So just one other protein that we kind of named based on what we first saw, but it can apply to other cells that are not epithelial. CD163 is also decreased, as well as IL-4 receptor alpha, or CD124. We do see some slight changes in the granularity and cell size, but it's not as dramatic when we look just by bright field images. And so with that, we've continued to focus on this. And this is work from Raekwon Williams, who was a high school student in the lab and in this program called Ingenuity. He's now a Goldberg Scholar at UNC Chapel Hill, doing his freshman year, given all of this. So he's hanging in there, and he's, social distancing and everything like that. And Fan Shin, who's my lab manager. And so Raekwon came to the lab and was interested in amino acids. So the glutamine project was going to be awesome for him. And very simple experiment. Let's take glutamine away. Let's look at macrophage CD163 levels and see if there are changes. And this is the blot that he was able to, consistently reproduce. And so we've continued to look at transcript level, which is the opposite of what we see on protein. But then when we look at CD86, we see that there's a decrease. And Fan has continued to follow this up by looking at cytokines that are altered when we take glutamine away, just using RPMI 1640 without glutamine. And we've also done the reverse experiments where we give glutamine back and we see these things change. I just thought it was too long of it would make the talk too long to include all of that data. And so what we're doing also, and we're doing this, we're going to do this in vivo with our syngeneic prostate tumor line, is use this prodrug of Don. So this was developed by Barbara Slusher and Jonathan Powell here at Hopkins, where this is Don, which is a glutamine antagonist, very toxic, and as an old school drug, it was used in the 50s, very toxic. And so what Barbara Slusher group, who has some of the most sagacious chemists on campus, they created this into a prodrug. And so this can be given into tumor microenvironments with high levels of cathepsin, which I've shown you that by mass spectrometry was one of our top hits, and into tumor microenvironments. And then these parts are cleaved out by cathepsin, and Don is in that tumor microenvironment instead of being all over the body like it was back in the day, as they say. So this is called the prodrug 5C, and they published on this in Science in November of 2019. So we're working with them. I'm not one of the people who does a lot of press, but all the work I showed you was in the K-22 grant, and we were selected to be in the annual plan and fiscal year budget proposal that is going to be presented to the United States Congress, so they'll put more funding into cancer research. So that's on the NIH's and the NCI's page. And finally, PI3 kinase gamma. And so, and I'm also giving you where I'm getting my inspirations from, and this is other people in the field. So I told you about Barbara Slusher, Jonathan Powell, the Agios paper. We got interested in PI3 kinase. For one, I published on it in graduate school, and then two, there was a really cool paper from Judy Varner's group when I was a postdoc, and they looked at the gamma subunit. And so, you know, I had that project in mind, and they looked at it particularly in macrophages. And so we began to really start to investigate it. And our hypothesis was, what is its expression in prostate cancer? We know that there are a lot of TAMs in prostate cancer, but there are some reports, or maybe it's expressed in tumor cells, we didn't know. And Judy Varner's papers didn't look at that. And so we began to look at gamma based on this cartoon, which is a comment to Judy Varner's group's paper. I think it was in Nature, where gamma induces CEBP activation, which leads to immune suppression. It also inhibits NF-kappa B. And so some of the first experiments we did were to look at prostate cancer cell lines that had intact and loss of PTEN to see if gamma was expressed in these cell lines. And this is something that was adapted from Thomas Nerschel, who's a grad student in my lab, but for a long time he was the lab manager of Chuck Drake. And so we looked across cell lines using macrophages as our positive control. And what we can see is gamma is not expressed in these cell lines, even when we use G protein coupled receptor stimuli to stimulate gamma, which is shown here in this cartoon. So it's not a PI3 kinase that's activated by receptor tyrosine kinases like the alpha and beta subunits are. And so then we look by message. For all the cell lines that I showed you, we don't see any message change in these cell lines. And so with all of that going on, simultaneously we were developing a syngeneic allograft tumor model we call B6Cap. It grows in the Black 6 mouse model, and CAP is cancer of the prostate. And this was derived from the Charles Sawyer model, the FVB on the FVB background. And so, so this shows that morphology. So this is the high Myc model, where it's cytokeratin A positive, AR positive, and Myc positive, which are canonical markers of prostate cancer clinically. And in the B6Cap model, they are cytokeratin 8, AR positive, Myc positive, ECAP positive. Important because it's an epithelial cancer. It's a adenocarcinoma. Arises from luminal cells. Cytokeratin 14 negative, NKX negative, synaptophysin negative. This is a pancreatic insulet as our positive controls shown here, and this was published in 2019. These are also, tumors are also sensitive to androgen, but can grow and become castration resistant. And so when we do intracardial implantation of tumors, or sub-Q allograft of these tumors, they can metastasize to the bone. And these are some of the images shown here. Notice that these are osteoblastic lesions. Most cancer cell lines, if you put them intratibial implantation, such as PC3, they're osteolytic. That's not what you see in human disease. And so we've, of course, investigated the immune infiltrate of these tumors, and IBA1, CD11B, and F480 are all macrophage microglial markers, and you can see that there are macrophages in these tumors. We've consistently looked at this by flow as well. And then we looked for CD3 positive cells, and again, a paucity of these cells in this tumor type. And so Tom, this is his PhD dissertation research, has been looking at white blood cells, and these were from the B6Cap Tumors sub-Q, just by transcript level. We've done Western blot since the lab has reopened in June 19th. And so in any case, when we sort CD45 positive cells and isolate RNA and do quantitative real-time PCR, there is transcript of gamma in these CD45 positive cells. When we do that for the CD45 negative cells, ideally are just the tumor cells and other non-immune cells, you don't see gamma transcript. And we get a lot of human samples, at least before things shut down. We performed some experiments. I think this is an N of 5, where we looked at human radical prostatectomy tissue, isolated white blood cells by flow sorting, and then looked at transcript level of gamma. And so in a lot of these radical prostatectomies, we get normal adjacent or benign tissue, and that is classified by the pathologist, and so we know that it's normal prostatic tissue. And we also get pieces of tumor tissue that's also called by pathologists, and we are able to isolate CD45 positive cells and then look for transcript. And what we can see, CD45 negative cells from the normal adjacent tissue have very low levels of gamma. The CD45 from normal adjacent tissue, the positive cells, do have some level of gamma, which we would expect, but the tumor tissue has even higher expression transcript, and these are across the equal amount of cells. And so we're working with Infinity Pharmaceuticals, who developed the gamma inhibitor IPI 549, as well as Judy Varner on the gamma knockout mice and flox mice. And so the conclusions of what I've talked about today, specifically the first set of experiments which was published in Science Translational Medicine about RP182, is able to target CD206 macrophages. In that model, we used a pancreatic cancer model as well as MC38 and other syngeneic models, and we saw that there was an increase in anti-tumor immunity after macrophages were targeted. We are also using glutamine deprivation to alter M2 macrophages into M1s, and we evidenced that by cytokine secretion markers, and we're doing some seahorse experiments there. We're looking at gamma, specifically in our human prostate cancer cell lines. It's not expressed, but in human tumors, white blood cells specifically from human tumors and from our novel mouse syngeneic line, there is gamma expressed among the CD45 positive cells. And so I don't work alone. I'd like to acknowledge my group. The work I've shown you today is of Debebe Tedros, Fan Shin, Tom Nerschel, and Ray Kwan Williams. Manali is also working on some of the seahorse experiments, and hopefully we can have that submitted soon. I have a very diverse group, and I'm proud of that. Everybody brings a unique set of skills from high school, undergrad, graduate students, and postdocs. So I encourage others to seek to have a diverse group and make it a priority to train underrepresented minorities and women in science. I have a lot of colleagues that I'm thankful to have: Jonathan Powell, Drew Pardoll, members of the urology department who have helped consent and provide surgical tissues from surgery so that we could do the experiments. My external advisors, Judy Varner at UCSD, who's helping us a lot with the gamma story, as well as Chuck Drake, who I believe is now at Janssen, from what I heard. And also the organizers there for the invite to Denver. I'd like to thank my funding sources, the Prostate Cancer Foundation, BKI within the Cancer Center, the K22 Award, as well as the Patrick C. Walsh Prostate Cancer Fund. And with that, I'll take questions. Thank you.
So, I wish you would hear the big round of applause. That very, very, very nice. Thank you for that. We have a number. So how about this for organizing the questions? We have a lot of questions in the chat box, and why don't I call on people specifically, and if you don't speak up, I'll just read it. The first person is MJO1 USB.
Oh yeah, I can see it. So are your anti-M2 drugs functional against both high and low CD206 positive macrophages? Yes, so that was done, that wasn't done in our group. That was done down at NCI, where they overexpressed 206 at certain levels in, I think, THP1 cells. That's in the paper. Our role was to, they wanted a primary human cell line that they could test the RP182 on.
Very nice. And Haida, are you there?
Yes, I'm here. So I was just wondering, because there are normal cell types that are expressing CD206. So is there any concern about toxicity with respect to these inhibitors that would target CD206 positive cells?
Yeah, so I'm curious. I mean, I know dendritic cells have some levels, M1 cells have some levels of 206.
So I was reading also sort of cells in the skin, forgetting exactly the cell types, but there are other cell types. I don't know how highly they express it, so maybe there's a therapeutic window. I don't know.
Yeah, maybe I would think that those cells, they're in the epidermis.
Mm-hmm.
I have to look into that. I wasn't aware of those, but I know that the resident macrophages of the integumentary system do have 206. The toxic. Well, I can't speak to the toxicity. I wasn't on that arm of the study, We didn't see toxicity in mice, and
Right, so
that was
good
the extensive.
indicator. Yeah.
That was the extent of our involvement with the manuscript.
Mhm.
So I'd like to piggyback on that. So the mannose receptor is also a receptor for a lot of viruses, isn't it? Like flu and
Yeah. Yep.
dengue virus. So have any of the do any of the antivirals target mannose receptor, and would they be useful in targeting metastasis?
So most the antivirals I know about are nucleosides. This is just a small peptide, and the mannose receptor, it binds to anything that's highly manicylated. So that's usually foreign objects. So your macrophages can recognize it and devour it. So I'm not sure if antivirals are the nucleosides are anti-CD206 drugs. That's I'm not aware of that.
Yeah, it'd have to be pretty. A lot of things would have to align for that. Sid, are you out there?
Yep, that was excellent talk, Jelani.
Thank you.
Really enjoyed it. So my question is more towards the sort of the lineage of these macrophages, and I don't know if there are anything known as prostate resident macrophages, but are the populations the same in your mets and the primary site? I mean, do they? I think that what we have more interest is, do the macrophage traffic with the tumor cells to the mets? And when they populate, I don't know if people know whether these are actually coming in from the tissue resident populations or which originally come from the yolk sac, or they're all bone marrow derived.
So in the normal adjacent tissue that we stained, we did see some 206 staining. I think it was like 12 cells or something. So there are perhaps tissue resident macrophages in the prostate, very understudied. But as the disease got worse, we saw increased stains of CD206. And even before we did any experiment, when I was in high school, there was a paper published that androgen deprivation therapy increased CD206, CD68 staining, excuse me, in biopsy tissue. That's a 2001 paper. And so we know that, or we believe that these cells may traffic from the circulation as monocytes and get into the prostate gland to do wound repair. Androgen deprivation therapy causes destruction and apoptosis of luminal cells. And so that causes a fire. The firefighters have to get in and put it out. The firefighters I'm referencing here are the wound healing macrophages. So we think that the ones that get into the prostate there may be the few resident macrophages, or they are monocyte derived, get in, differentiate, polarize into M2s, regulate IL-10 and other pro-metastatic fields to repair the wound. And as far as seeing them traffic with the Mets, so John Condilis was on my short list of people I wanted to postdoc with because he had the videos of that when I was a graduate student. So he has published on MENA 11 and shown in breast cancer that macrophages do traffic with the tumor cell. And they have, I mean, they're on YouTube now, but they had published it in one of the high tier journals when I was a graduate student. So there's data that they co-localize with tumor cells and traffic together. Do they traffic to the lymph node? Because they use like an artificial angiogenesis tube that I don't know if that's been tracked and traced efficiently to make that call. But we do see them in the secondary and terminal sites. in high numbers. And we also see them in the rapid autopsy tissues from men who died of prostate cancer, unfortunately.
Nice.
Jill, can I piggyback onto that, Jill?
Oh, sure. Yeah, yeah.
Okay. Yeah, my question was kind of related to that down at the bottom. I think I was also wondering if it was the androgen deprivation therapy and the sort of trauma that causes that increases the CD206 positive macrophages, and you kind of answered that, but I was wondering if you looked at any biopsies of, prostate cancer primary before treatment compared to after treatment with enzalutamide or bicalutamide or any androgen deprivation therapy to really nail that down.
Yeah, so we want to go back and look at some of those samples from the TMAs. I think only a few of the men had bicalutamide, and those were the men who presented with Mets or those who were castration-resistant. I can go back to the slide if you like, but those numbers weren't substantially high compared to the other samples. Can you see the slide here?
Mm-hmm.
Yeah, so in the orange bars, and also this distant Met was in IHC was castration resistant. These are just like representative of the 200.
Oh yes, so it goes a lot after therapy. Yeah.
Yeah.
A really nice talk. Beautiful work. Thanks.
Thank you.
Michelle, are you there still? Michelle Williams?
Yeah, I'm here. Can you hear me?
Yes.
Yes.
Okay, good. Thanks for a great talk. I was wondering, there's some evidence, I think it's from use of CSF1R inhibitors that suggests that if you switch the ratio of M1 or M2 to M1 to favor M1s, that might actually be better than just getting rid of all the M2s in the tumor microenvironment. So I'm wondering how this might play into your different approaches of targeting the macrophages, like with the RP182 versus the glutamine deprivation.
Yeah, so the great question. So the RP182 is thought to be kind of like a strategy to kill the macrophages or destroy them to promote anti-tumor immunity. So you can use anti-PD-1 or some other therapy in combination.
Mm-hmm.
I think the glutamine deprivation is somewhat coterminal with the CSF1 receptor inhibitors. I think that Roche has developed where they can switch the M2 to an M1 and lead to recruitment of lymphocytes to that tumor microenvironment. I emailed, I forget who, in like 2015 trying to get some CSF1 receptor, and there was a lot of red tape with it. So we ended up trying to do something else. But yeah, I'm a fan of it. I know it targets both types of macrophages, though I think, at least the data from what I remember, because the CSF1 receptor is on M1 and M2. And we wanted to just try to target the M2s more specifically, like using 206 or glutamine, because M1s don't use glutamine the same ways M2s do.
Okay, thanks.
Phil.
Hey, that was a fantastic talk. My question was about osteoclasts having sort of shared mechanisms, and they're CD68 positive and can be CD206 positive, and you could have sort of an unintended or maybe intended benefit of inhibiting osteoclasts from resorbing bone and strengthening bone in some of these patients that had osteolysis or even mixed lesions in the bone. Have you guys looked at some of those models or looked at any of the bone quality to see if osteoclast number activity was affected?
So the RP182, so great question. You must work in a bone lab, I would guess, or you just read the bone papers.
Maybe.
Or you're part of, or you're an honorary member of Bone Thugs and Harmony. Or
all the above.
Right.
At the crossroads.
So we did not look at 182's effect on osteoclasts in the bone, but that's an excellent question, and that's something we should definitely do, because osteoclasts are basically bone resident macrophages.
Absolutely. Thank you.
Yep.
Steve?
Yes, hi, this is Steve Nordine. I'm semi-incognito there, I guess. Um, just one quick comment. Also, for the old timers like me, Angelo DiMarzo did his MD PhD
Yep.
here at this university. My question concerns the drug CB839, which is approved for use in humans. It's a glutaminase inhibitor.
Yep.
Have you looked at the effect of that on your M2 cells?
Excellent question. So we have some of the data was inconclusive. We were on the CB839 from Calithera Biosciences because the person over the study, Susan Demo, is a Hopkins alum, and I met her at AACR. So I tried it as a postdoc, and some of the data was inconclusive. And then I know she worked with Jonathan Powell because of the importance of glutaminase on lymphocytes. I don't know what their data was, but we did look at CB839 before it went to clinical trials and all of that. Yeah.
All right, thank you.
Mm-hmm. I think also the Calithera Bioscience CEO and her husband are also Hopkins alum as well. I can't remember their names right now, but I know Susan Demo did her PhD at Hopkins.
Scott, are you still out there?
Yeah, I'm here. That was a really nice talk, and I think you already answered my question. It was really what is it intrinsic about the tumor that is driving the M2 increases?
Yeah. Go ahead.
Yeah, that's it. That was my question. Yeah.
Yeah, I think it's tied to the old adage. I don't know who's accredited with saying it first, but it's that a tumor is a wound that doesn't heal, and so it's going to consistently recruit wound healing cells to that site, and that is why you're going to consistently see, these wound healing cells, whether they be, these MDSCs that folks are studying, or other wound healing cells at the site of these tumors. It's also present in like ischemia. So Lou Cantley, who discovered PI3 kinase, I bet you didn't know that his brother was a nephrologist who postdoced with him and studies ischemia and looks at M2 macrophages. I can't make it up. I think I impressed him when I met him, and I talked about his brother more than I talked about him. His brother's work. But yeah, you see it in ischemia. You see it in all these types of wounds, and you also see it in like Hellman's infections. That's the only like disease that you see it in. It's not prevalent in the United States. So it's in metastasis and ischemia and like Hellman's infection. But I think it's tied to it being a wound that doesn't heal. I don't know who's credited with saying it. May have been Aristotle, Hippocrates, Imhotep. Somebody said it before me.
You might have said this and I missed it. Did you look at different metastatic sites, and was there a difference? So, if you have blastic lesions, you might really think that those are wounds really, because it's abnormal bone that's being laid down. It's not normal bone. And so are they more attractive for the M2s versus the M1? And I missed if you said it already.
Yeah, so I'm on the rapid autopsy series slide. So we've done about 30 cases from multiple sites. I'm only showing epicardial lymph node. We have the first right rib. We had the tibia. We had the femur. We had other soft tissue mets, and all of them are high for 206 and 163, or 206 alone by IHC or by flow. So we've looked, and these are across multiple patients over multiple years. So these studies started before I even came to Hopkins, and are still continuing. So we do see high TAM infiltrate at multiple, metastatic sites in different patients. Now they're not all age matched or anything, but we do see it at multiple sites.
Brian Haugen, are you still out there?
Yeah, thanks so much. That was really fantastic work and a fantastic talk. I was thinking with the glutamine and just thinking a bit more broadly about cancer metabolism. And have you taken a look at potentially the role of lactate in this model?
Yeah, so lactic acid is something we're interested in. We have not looked at it. We've looked at like iNOS and arginase, and I know that ties together with it, but we have not looked at lactate. It's something we want to do.
Very good. And James.
Great talk. Really enjoyed it. And I would definitely second what you said about lab diversity. It's not just the right thing to do. It actually makes for way better science and potlucks.
Oh yeah. So my lab is like from like 6 different countries. I forget how many languages people speak from, different cities, different experiences, different ages. It's just something that I always said I would want to do. And to be honest, like when I was at Michigan State, I was the only Black grad student. I was the first Black grad student. I thought, what a shame. This is a great school. And this program, was founded in the 80s. I was born in the 80s, and they hadn't had any Black graduates. I thought that was astonishing. But I think we all could do a better part at it. And Cell and other journals have put things out about trying to address diversity in the pipeline, and it's on us. A lot of women of all ethnicities and racial identities fall out of the science pipeline, proverbial pipeline in high school because they're disinterested or they don't see enough women, and so we all can do a better part at that for sure.
Absolutely, yeah, at all levels. I mean, we need to not just get more students in, but we need to have diversity at every level and promote people all the way through. So my science question, I'll follow up on that, is that, and this kind of follows up a bit on what Brian and Michelle were just asking. So it's interesting that both your peptide that binds to the mannose receptor and glutamine deprivation both can cause the switch to an M1 phenotype. And I'm wondering if you've looked at metabolic pathways downstream of those two changes and whether there's some sort of intersection. In other words, are there some commonalities between those, or are they just two different ways of ending up doing the same thing?
Yeah, so we have looked at caspase cleavage. We don't see caspase cleavage when we take away glutamine. We do see it with the RP182. And there are other peptides that this company is making to look at some other aspects of M2 biology, but just simply removing glutamine doesn't appear to activate caspases. We are looking at metabolic profiles, so we're doing seahorse experiments now, and we're also looking at the ability of exhausting or ability to kill T cells or to have similar phenotypes like secretion of certain cytokines as T cells, and doing T cell co-culture assays with cells that have been treated with DON or BPTES or without glutamine. So we're trying a lot of different things now. Now that the labs are back open, we have better ideas.
And that's always a little bit of a difficult thing. For example, glutamine deprivation isn't just going to switch your macrophages, but it also deprives the T cells of the needed glutamine.
Right.
So,
yeah.
And so that's why I find the peptide result much more exciting, because it may be a more targeted approach.
Yeah. And we have gotten that comment a lot, and that is also the reason we wanted to use the GHU83 in vivo. So we're doing all the glutamine deprivation because it mimics somewhat of how this DON prodrug will work. So the DON prodrug, now Jonathan Powell is a T cell metabolism guru, so they published that in Science in November. We want to try it for macrophages because they have a similar M2 macrophages have a similar dependence on glutamine as lymphocytes do. And we got funding for the K Award to do it. So that's another reason. So we want to test it there. and we're also doing some heavy carbon tracing experiments as well to look at a lot of the, you know, metabolic profiles and the TCA